Welcome back. This is part 2 of translation in proteins. Again, I think some of this is going to be familiar to you. I think some of it is new. Come at this with the fresh student mind so that you're learning it all again, and then see how it fits with what you learned before. I want to stress here that this is stuff that we are on the forefront of what we understand. Sometimes this information changes, and that's a good thing. We're understanding it better, but some of this may not fit with what you learned in Bio 1. This textbook that we're using for genetics is pretty up to date. It will actually update again next year, but it's more up to date right now than the Campbell book you used in Bio, just because of that the volume of information in Campbell, not because of anything the publisher is doing. We know that our understanding of translation comes from bacteria. It is much easier to study in a plate of bacteria than it is to study inside of a living eukaryotic organism that is multicellular and doing different things in cells at different times in different parts of its body. So eukaryotes, some of them are single-celled, some of them are colonial, and some of them are multi-celled. The ones that we are most interested in as humans are really freaking complicated. We're mostly interested in humans and cats and dogs and farm animals. That means that we have less understanding of the things that we are most actually interested in. We do know that transcription and translation in eukaryotes are separated by location and by time. There, in a bacteria, there can be simultaneous transcription and translation because there is no nuclear envelope. In eukaryotes, the nuclear envelope separates those two processes literally by a barrier. You transcribe, then you cap and tail and take out all of the introns and splice together the exons and then export the final mRNA to a ribosome, which is in a different location in the cell. The ribosome for the eukaryotes is larger. It's an 80S ribosome. And it has a few major differences in the initiation process that I want to show you. That Shine-Delgado sequence is only in bacteria. We have a different sequence in eukaryotes. All of our initial understanding of translation comes from bacteria, but we start out right at the beginning by eukaryotic translation is completely different from the start. We don't have a Shine-Dalgarno sequence. What we do have is cap-dependent translation. Bacteria do not do the capping and tailing. They don't have introns and exons. They make their mRNA sequence, and then they execute that mRNA sequence into a polypeptide chain. We have all that in-between step where we do the modification of the mRNA before it leaves the nucleus. A big part of that was putting on the cap and the tail. That 5' cap is essential in binding to the ribosome and placing that mRNA in the right orientation and also getting the process started. As the cap binds into the small subunit of the ribosome, the cap slides along and eukaryotic initiation factors, EIFs here, bind to the cap side. And then the first tRNA that recognizes the site to start the process of translation is a unique tRNA. It is still using the same amino acid that we use in bacteria, but this one is only involved in initiation, this particular tRNA. It's used at the Kozak sequence. This is Mary Kozak has discovered this more recently. I'm thinking the 1980s, if I remember correctly. This is a purine. It's 3 bases upstream from the start codon, and then that start codon is followed by a G. So I have an A or a G, an adenine or a guanine, a purine, and then there's, I don't know, whatever base, it doesn't matter, followed by an AUG, G. This is my start sequence. I put these question marks here. In the book, I think it uses N's, which I thought, although standard, is confusing. It means it doesn't, I don't know what this is, and I don't care what, nucleotide is here. I have 2 whatever nucleotides followed by the start codon and a G. Remember that poly A tail, we've used it in chapter 20 to reverse transcriptase something. This poly A tail is also used as the mRNA leaves the nucleus to kind of mark that it's finished, to keep it from being degraded. It is also essential in the translation process. At the end of the mRNA transcript, this running off all the end is also bound to an elongation factor. This elongation factor, EIF4G, binds to the one that bound to the cap, and it creates this loop of mRNA. This is called closed loop translation. I don't have a great answer for you as to why we do it this way, but we definitely do it in a closed loop, so this becomes a cycle of translation rather than just a long piece that is mRNA going off into the distance. And kind of this is a good place to point out, and I think I did earlier in a couple of the lectures, we're getting into material that we are just starting to understand. Some of the things, including how this process works, is not the same as you learned in Bio 1. For some of you, maybe some of you took it very recently, but for most of the things you learned in Bio 1, a couple of these little translation bits are really different. We have just discovered that some of the enzymes that you may have learned in Bio 1 actually don't do anything like what we thought they did. The ribosome is much more of a ribozyme than we thought. This is super cool. This is, you are learning stuff that is at the forefront of what we understand for how life works. And that means that I may be teaching you this year, and next year I'm going to have to rewrite all of these lectures because we find something new. This is where it gets really fun. And I think that's why you're here, is more to learn how you can find out more about how the world works. I think this is kind of a fun part in this course in general, where we've stopped learning what they did 100 years ago, and now we're like, what do we know now, and what does that mean? And how can we use it in different ways? And how are we wrong? It's pretty cool. Our understanding of the consequences of this, and most of the rest of translation, is really similar to bacteria. So I'll leave that for you to just insert the fact that initiation is very different in eukaryotes and prokaryotes. The next thing we're going to do is we have these proteins, right? We made the protein, now it has to function. Our understanding of the process of translation and transcription comes from inborn errors that are mistakes in proteins. We can't see with our eyes the process of translation. We can get a general idea that it's going through, we can get the shape of it, we can take pictures of ribosomes doing little ribosome things, but we can't see the sequence it is reading. Those molecules are really, really small. The way we understood this is through mistakes. So inborn errors means genetic errors that resulted in expression differences. There are two diseases I want to introduce to you that you may have actually already know about, but two diseases I want to introduce to you as part of this that are really instrumental in our understanding of these processes of how proteins are made. AKU is black urine disease. Alkaptonuria, this is urea, this is in urine. It's black urine disease. It is a failure to metabolize a single protein and an acid. It's not fatal. It causes your urine to be black. We identify it very early. When babies have black urine in their diaper, we know that as scary as it sounds, we know that they are expressing this inability to metabolize a particular molecule. And if you're really feeling spunky here it is. This is a metabolic cascade. Phenylalanine is converted into tyrosine, into 4-hydroxyphenylpyruvic acid, which is converted into homogenistic acid. This part here, if you cannot metabolize it, you check out of the metabolic process here and you have black urine. If you do metabolize it, you continue on and you don't have black urine. This is an easily observable failure to metabolize something that is caused by a protein. This was one of the first ways we understood it. But if you know a little bit more about bio, you're also seeing something along here where there's another block up here, meaning that it's a failure to do something. And this word may be actually familiar to you as well. PKU, maybe you have it. I don't know if there's one in so many. No, I actually make, in my Bio 1 class, I make them calculated. I can't remember off the top of my head. It's a substantial number of people. It's enough people that we can, we label everything in the US with warnings if you are PKU positive. It is the same metabolic pathway. It is the same pathway of breaking down this phenylalanine, which is really an amino acid. If you are unable to break phenylalanine into tyrosine, you have PKU. That's not a P, PKU. This is a little bit more catastrophic. If you have this, what happens is you build up phenylalanine in your cerebral spinal fluid. This causes blockage of signaling and also your ability to clean up and maintain your brainstem and your brain. This can lead to severe intellectual disabilities. Phenylalanine here, I'm going to do it here, is really common in artificial sweeteners. In regular food, it is often not independent. You have to be somewhat careful, but if you don't eat any manufactured food, PKU may not be a big issue for you. But most artificial sweeteners contain a phenylalanine on the end that you then can't metabolize. If you have this inborn error in your metabolism, and this will cause long-term damage. So we get around this as a human society by basically labeling all of it. If you look at your anything with artificial sweetener, on my gum, my chewing gum has a mark on the back of it. We identify this very early. It is a test that we do literally at birth. They take the baby away from you as soon as it's born. They do a blood stab on the bottom of the foot. They wipe a little smear of blood on a card, and if it's positive, then that kid is and you as the parent are given nutritional advice, and so that you can avoid it for your whole life. It's a fun thing in genetics. As we go forward, we'll see if we have time. I do it in Bio 1, where you can actually predict the incidence of people with PKU because it is not subjected to any sort of natural selection pressure at this point, because we educate everybody with it and they avoid having any sort of damages from it. So it persists in the population at exactly the same rate. It's an interesting way of looking at population genetics as well. Also, how we understood this. We also had a progression of ideas of what exactly is a gene. Initially, the assumption was in the 1940s that it was one gene for one protein. Well, that didn't play out. And when we started understanding that many proteins did a lot of different things, and that proteins could be made of enzymes. Enzymes are catalytic molecules that are made out of amino acids that cause reactions to happen at a lower activation energy. They cause reactions basically to happen faster with less energy input. Experiments on the pink bread mold, which we've seen in a couple places, Neurospora, it led to the idea with Beadle and Tatum's experiment that we did before that each gene codes for one particular enzyme. They knew that wild pink bread mold could digest everything that it needed to and do all of its metabolic processes. And they knew that if you zip living things with UV light, it causes the degradation of the DNA. It actually causes the molecule itself to become misshapen and break and reform in ways that maybe don't spread information accurately. If you have a mutation, then you are, as a pink bread mold, unable to metabolize some of the substances or make some of the substances you need to live. So what we did in previous experiments in other lectures, we then provided them with those substances. We had nutritional mediums that we put them on, and we provided them or didn't provide them, and we could select to see who could survive on this medium. If you are a completely normal, non-mutated bread mold, you can survive on every kind of medium. I can give you a completely unnutritional medium and a minimal medium, and you can survive fine. If you are a mutant, you are unable to survive on medium that don't provide the things that you can't make, which makes sense, right? If you can't make it, you need to get it. So what they did, Beadle and Tatum, is they used a series of mediums, places where the bacteria, or in this case, the conidia of the fungus, a conidia it's just spores that are being reproduced asexually. They use a series of medium to select out each different kind of mutant. They would expose a fungus to UV radiation, initiate a mutation, and then see how many mutations did they cause. Well, what they determined was that every time they caused a mutation, it affected the ability to digest one particular substance. They assumed that that meant that every mutation affected one part of the DNA. One part of the DNA represents one gene, one enzyme, one gene, one ability to digest something. So you know that this doesn't make sense. Coming from a 21st century world, we know that some enzymes are actually comprised of more than one polypeptide. You looked at things like ATP synthase in Bio-1. That's a catalytic enzyme that manufactures ATP. It's got a lot of different structures in it. Collagen, hemoglobin are all tertiary, fourth level proteins, which means they're assembled from multiple polypeptide chains into a functional unit. I'm going to use hemoglobin here because I think it's familiar to everyone. Hemoglobin is the protein in your red blood cells that carries oxygen and iron. It's made up of 4 polypeptide chains, 2 from one gene and 2 from another. So it uses each gene twice to assemble a 4-part machine. Linus Pauling and his colleagues showed that different parts of the hemoglobin actually moved at different rates during electrophoresis. This is why I wanted to do 20 before 14 here. We're going to do electrophoresis, but not like this. He's doing it with proteins. When he did this, he found that different parts of the hemoglobin moved at different rates. That implies that they are different sizes. So there are different parts of a hemoglobin that are different sizes. And when you have a mutation in one part of a hemoglobin that causes that entire hemoglobin to function differently, we actually see that that mutation only occurs in one of those parts of the hemoglobin. So does that make sense? I feel like that was convoluted and doesn't need to be. He found that a mutation in one polypeptide chain only affected one polypeptide chain in the hemoglobin and didn't affect the other parts of the hemoglobin. The classic example for this is sickle cell anemia, which is what we're actually looking at here. Sickle cell anemia is a single point mutation. One nucleotide is different. That nucleotide in that particular polypeptide set of instructions, that gene, causes the wrong amino acid to be inserted. That causes the shape of that part of the hemoglobin to be not as expected. Vernon Ingram later showed that that is actually in the globulin part of the hemoglobin. That change in amino acid sequence results in the red blood cells being sickle shaped. A sickle is like a little cutting blade, right? A little cutting blade. Needs to be round in order to be effective at carrying oxygen. People with sickle cell anemia that are expressing it have 2 alleles that are both incorrect. Those alleles cause the red blood cells to have one single misfolding error, which causes those red blood cells to not be able to carry oxygen. They can't bind to the iron, and they can't carry oxygen as a result of that. Fingerprinting them, these sequences, actually looking at how they moved through the electrophoresis medium, is the way we identified the fact that they are just a little bit different. They're different lengths, they're different sizes, proteins in this cases, and they move through the medium differently with the electricity. So I want to step back into Bio-1 here for a brief second and make sure we're all on the same page. When I synthesize a polypeptide with the ribosome, I am not done. That polypeptide chain is a primary protein. It is held together with peptide bonds through basically dehydration synthesis that is done in the ribosome by the ribozyme in the A site of the ribosome, where I catalyze the formation of a peptide bond. The chain of polypeptides that comes out of the ribosome is not functional yet. Proteins are only functional if folded. They are folded in at least secondary and tertiary structures inside humans. Secondary structures are when the polypeptide that comes out of the ribosome, basically the cell is very small and the ribosome can be catalyzing a fairly long polypeptide. It gets bumped into itself and it forms hydrogen bonds between the amino and the carboxyl side of the amino acid. It forms little hydrogen bonds holding the amino acids together. They're strong enough to hold a structure that is either a loop-de-loop, an alpha helix, or a pleated sheet. So an alpha helix or a beta pleated sheet. In humans, I don't know of any protein that looks like this as a secondary structure. My understanding is all human proteins are folded at least once more. This is the structure that holds together something like spider webs, though. They're very fragile. They're held together by hydrogen bonds. They're strong enough, though, that they can function as a structure, but not strong enough that you can't poke them apart with your finger. Tertiary structures, so this is secondary is 2, tertiary is 3, or the third level of folding, is where I take all the amino acids in my polypeptide chains that are kind of being structurally held together by hydrogen bonds in maybe a folded form, a lightly folded form, and then I use other proteins to forcibly or spontaneously, depending on the structure of the protein, interact the R groups of the amino acids together. So not the consistent structure between all amino acids, but all of the little variability that makes one amino acid different from a different kind of amino acid. I take these little proteins and they're going to chaperone folds and interactions, everything from covalent bonds to hydrophobic, hydrophilic interactions to sulfur dihydral bonds, a whole bunch of different kinds of bonds that holds a three-dimensional, really twisted shape of my protein. And now these proteins are, in many cases, functional. Proteins function by having a space in them, an active site where they can interact with other molecules. We literally put them together more like Tinker Toys or like Lock and Key than just a chemical reaction. Having this three-dimensional shapes gives them the shape that they need to do their function. But there's one more possible structure. I can take my folded tertiary polypeptide chain and merge it together with another folded tertiary polypeptide chain. This is hemoglobin. Hemoglobin is made of 4 polypeptides. Each of those polypeptide chains is not primary, it's not secondarily folded, but each one of these, the beta chain, each alpha chain is individually folded into a tertiary structure and then assembled into the 4th level of protein folding, quaternary, level 4 structure, where they are multiple polypeptides forming together to make one functioning structure. The one gene, one enzyme idea falls apart at this point when I start looking at the complexity of the fact that in order to have a functioning protein, I need multiple genes worth of polypeptides in order to make it work. When my folding process is done, I often do a couple little steps, and I want to point this out, that I can modify my proteins after they've been folded. I often remove the amino acid on the N terminus, the one that has the amino, the nitrogen holding side free, or I acetylate it. I add an acetyl group to it. Some of the amino acids are modified during a post-translational modification. I can either activate them or deactivate them, put a tag on them. I can trim my polypeptide. I can create a protein at this point that is not active until I do one more step to turn it on. This way I can store a lot of proteins without having to worry about the fact that they might be hydrolyzing or breaking things down. The things that help me do this are called chaperones. Chaperones are protein that aid in the folding of other proteins. If you really think about this, it starts to get bonkers really fast. In order to make this hemoglobin that you cannot live without, I need at least 2 genes. I need to make 2 copies of each one of those. So I need 4 polypeptides. I've got to assemble them correctly. I need all my chaperones to actually be working. So as I make those tertiary and quaternary folds, I need to make sure that those other proteins are also shaped right. And I also have all of the genes for those proteins functioning. And I've got to have all of the enzymes that are going to acetylate or cut and clip and make this into a functional unit. And then I'm going to export it out of the cell using a bunch of proteins that push it out in an exocytosis. Holy cow, the fact that anything works at all is amazing. There's so many steps in each way. Each time you process one of these things that are actually essential steps. And I think it's amazing that we work at all. These post-translational modifications, if they go wrong, it's really bad. So the fact that it works right all the time is amazing. But of course, there's always a chance that everything goes wrong. If you misfold proteins consistently, it can be you just not making the thing that you're supposed to be making. It may mean that you're making incorrect hemoglobin and you have sickle cell. Or if you're creating free misfolded proteins, there's other options. Prions are misfolded proteins. Your body is making proteins that are folded wrong. Some of these can initiate the replication of themselves. So we're getting into microbiology territory here. Creutzfeldt-Jakob disease, scrapie in sheep, mad cow disease, which is BSE, all result from misfolded proteins. They tend to dramatically affect the brain. This is an MRI. You can see the proteins being precipitated inside of this. Then that means that they are interfering with how the brain actually works. We're still learning about prions. They're super scary. I think you did them in micro. There was like one little part of a chapter about them. We have a problem in New Jersey with deer getting a prion disease. And so if you see deer, it's immediately reportable. And you see deer bonking their heads against trees and stuff. That's actually, you need to call the state and have them come out because we don't want it to spread from deer to deer. It's a prion disease. So a little bit of practice on that note, a very happy note, on that practice note, a little bit of practice with me. So when we're doing elongation in translation, where do the tRNAs enter? I'm going to go forward, pause me if you need to. The A site, it's the arrival site. It is also the attachment site. All the stuff actually happens in the A site, all the good stuff. This is hemoglobin. We just saw it. What levels of structures do you see? And I know there's an E here. I don't usually have E because a lot of the professional tests only have 4 options, but I want to give you more thinking space here. All right, I'm going forward. Boop. You can't see the primary. You would have to see the amino acids, but otherwise you can see everything else. The folding within each subunit, each polypeptide, the secondary, tertiary, and quaternary interactions between amino acids. And finally, a little bit of practice with me. This is, you probably are going to have to pause me. This is in your the post-it version of this. The answer isn't. You're trying to recreate Beadle and Tatum's experiment here. You have your spores of Neurospora, the pink mold. You want to induce some mutations. And although you know that minimal medium will grow normal spores, you find one that doesn't grow on a minimal medium. When you inoculate, so you put it onto a supplemented medium, So you inoculate means you take it and you add it to a new and cause a new colony to grow. You observe these results on this side. From this, what can you conclude about your mutant? If you put it in a medium with everything on it, it grows. A minimal medium missing everything, it does not grow. Minimal with pyrimidines, it does not grow. It grows like gangbusters on a purine-filled one. It doesn't grow with just straight amino acids. It doesn't grow with adenine. So you have to go back and remember what auxotrophic means. And then what's wrong with your pink mold? I'm going to go forward. You can pause me here. It's missing purines. So in retrospect, super obvious. The only place it grows is if you're providing this. If you can't make something for yourself, and we have this in humans, right? We have essential nutrients that you cannot make yourself. You cannot make some of the amino acids in your own body. You need to get that from food. They are essential for your diet. This mutant can't make purines now. This mutant needs them provided for it. It is now an essential nutrient for it. It is auxotrophic for purines. With that, that's my end of practice with me. I will see you in lab this week. We have a couple fun things to do. If you are watching this first, and I put a note in there, make sure you take a peek before Thursday at the chapter 20 intro. We're going to do some PCR. We're going to do some restriction enzyme stuff. I'm going to talk about it in lab. And then if you have time, watch. There's like a 8-minute video of a guy doing our lab, slightly different procedure, but he has a lot of tips and techniques that are going to be really helpful for you to see ahead of time. Right before lab, or even as you get in, worst case, we'll watch it. But as you get in, take a look at that little video, because he has a lot of, those techniques are good lab techniques for getting around some of the problems with the lab equipment in every lab, not just ours. And I'll see you in class. All right. Have a good week. Bye-bye.